Scientists have isolated a specific biochemical sequence that triggers a cascade of changes deep inside the human brain. The brain normally functions as a highly regulated network, using billions of precise chemical messages to keep its internal systems in a constant state of balance. However, in certain forms of autism spectrum disorder, that balance is lost. Researchers found that specific parts of this cellular network operate in a state of unregulated overdrive, with biological signals firing far more often than they should. The system responsible for this hyperactivity is a control pathway called mTOR, a central hub managing cell growth and protein production. When mTOR loses regulation, its activity expands aggressively, overflowing normal limits and producing proteins at a rate that interferes with neuron communication. For years, the cause of this hyperactivity remained a mystery. Researchers needed to identify which specific internal mechanism was failing and allowing the mTOR engine to accelerate without limit. Mapping this specific pathway shifted the focus of the research. Scientists began hunting for the microscopic trigger capable of cutting the cell's biological brakes. To find that trigger, the team looked at the natural defense systems designed to prevent cellular hyperactivity before it starts. This schematic highlights a protective protein called TSC2. It acts as a biological brake pedal on the mTOR pathway, restricting activity to keep growth at safe levels. But examining hyperactive cells revealed a startling observation. The TSC2 protein was actively disintegrating. As the protein levels dropped, the brake weakened, and the mTOR pathway surged through the gap. Without the restriction of TSC2, the neuron loses its primary regulation mechanism. The rules that govern how the cell grows and communicates begin to fail. The consistent disappearance of the TSC2 protein across these samples pointed to a targeted biological sabotage occurring inside the cell. Looking closer at the cellular environment, researchers found an abundance of a tiny signaling molecule called nitric oxide. While it usually helps brain cells communicate, its behavior changes when its concentration rises. The team discovered that high levels of nitric oxide trigger a specific chemical reaction that targets the cell's internal regulators. Here we see that process, known as S-nitrosylation. The small nitric oxide molecule forcefully locks on to the much larger structure of the TSC2 protein, acting like a chemical tag. This specific tag corrupts the TSC2 protein and marks it for destruction. Once tagged, the protein is removed by the cell's own cleanup systems, effectively dismantling the brake. This creates a direct domino effect. Nitric oxide tags the TSC2 protein, the brake is destroyed, and the mTOR engine is left to accelerate out of control. With the chain reaction identified, the researchers set out to prove that this microscopic sequence could be manually interrupted. In the laboratory, they used targeted pharmacological tools to lower the production of nitric oxide within the affected neurons. The results in the laboratory were immediate. By decreasing the nitric oxide, the researchers prevented the tagging of the TSC2 protein. The biological brake rebuilt itself, and mTOR activity returned to a healthy balance. To confirm the link, the scientists engineered a version of the TSC2 protein that was immune to the nitric oxide tag. Even in the presence of high nitric oxide, this modified brake remained intact and successfully stopped the cellular overdrive. To see if this happened outside the lab, researchers examined clinical samples from children diagnosed with autism spectrum disorder. This split screen shows that the laboratory data on the left and the human clinical samples on the right display identically matching trends. These clinical samples contained the exact same biological markers, reduced levels of the TSC2 brake, and a corresponding surge in mTOR activity, confirming the mechanism exists in the human brain. While autism is a complex condition with many different biological drivers, mapping this specific chain of events provides a clear target for future research. It offers a new framework for developing treatments designed to restore cellular balance in the brain.